I hope you guys enjoyed the research talks yesterday. Today is also going to be some amount of research, but we're going to start with pathogen genomics of unknown pathogens. How do we try and look for them? Yesterday's talk by Ashwin was really nice. It gave us a deep history of microbiology and how we think about these things, which we tend to forget. Like, just like when, as say ecologists, when we start using molecular approaches, we just use them as tools. But we forget the history of it. And today we're going to start off with Chitra. So Chitra, Dr. Chitra Pattabhiraman is a PhD from NCBS. And for as long as I've known her, she's been really passionate about unknown pathogens. So she was an early career researcher supported by the DBT Wellcome Trust India Alliance. She worked at NIMHANS for that period. And then during the COVID, she did a lot of work on sequencing COVID, some of the first studies in Bangalore. And now she is a consultant with the BMGF, Bill and Melinda Gates Foundation, because they have such an interest in supporting work on unknown pathogens. So we're really lucky to have her, and thanks for joining us, Chitra.
Hi, good morning, everyone. Thank you, Uma, for that introduction. Thanks for inviting me to give this talk. Can you hear me okay? So I'm going to give you an introduction to pathogen discovery. And it's mostly going to be around viruses, because that's sort of what people have done a lot of, and not so much about bacteria and so on. But I'm going to give you a little bit of the history, and a lot of that has to do with bacteriology. I'm sorry if I'm repeating things from talks that you've already heard, but I'm happy, please talk to me about it, and I'll see if I can add more things or go faster in bits that you've already heard. So I think we're going to be discussing 4 cases, right? One is pathogens with outbreak potential, where you have a cluster of cases, you don't know what's causing them, okay? Let's call those with no known etiology. Can you think of where this would happen? Like in what kind of conditions? So for example, if you have a cluster of respiratory infections, a lot of human beings falling sick in an area, and you don't know, like all your routine tests are negative. Do you know of any case where this happened?
I think we all know, right? Anyone? Okay, a cluster of respiratory infections where we didn't know the cause.
Yeah, the start of COVID. So we're going to discuss that. Sorry if you have a bit of pandemic fatigue, but this is going to be different from things that you've heard so far, I think. Then conditions of unknown etiology, where testing doesn't reveal a causative agent, but we're talking about single cases here. So you have one child or one person who has fever or brain infection, usually, typically something life-threatening, but you don't know. And so you don't know how to treat that person. That usually comes under conditions of unknown etiology. And here we'll be talking about something called acute encephalitis syndrome, or AES. And basically, these are brain infections. And roughly, you can diagnose about 30% of them, okay? So we'll be asking the question, what about the other 70%? Is there something interesting there? Then, of course, we have animal die-offs when you have a large outbreak in animals. And I'm sure you've already heard about zoonotic potential and zoonosis. And so here we care about this, of course, from the animal point of view, but also about the probability of spillover and an evolving pathogen that is able to adapt to different hosts. Does anybody know an example of this happening right now?
Animal die-offs, yeah, we also discussed this, right?
Avian influenza, that's right. Yeah, so we'll discuss this a little bit. There is this exercise, and there's a lot of backlash and pushback against this now. And we should be talking about it a bit more, which is cataloging what's out there, surveillance of other hosts and animals. So these two are going to be more discussion points, because these are not areas that I work on directly. This particular one is what I've worked on the most. So the talk will also be sort of around that, right? So there are lots of pathogens. We have lived with them ever since the beginning. Since we have human populations, we have had viruses, and these also have spread as humans have migrated, okay? So this is Egyptian hieroglyphic, and it shows you a person who looks like that person might have polio. This is, I think it's called the Florentine Codex, this particular document. It's really old, I think 16th, 17th century, around that time. So this is when smallpox got transported from Europe to South America. And then you have these descriptions of how people who had not been exposed to that before now were incredibly susceptible to that. This, of course, is the Spanish flu. This is how a pandemic looked like in a hospital setting in the beginning of the 20th century. You might have already discussed, is this familiar now? Have you seen this graph multiple times now? Once. Okay. So this is the sort of basic framework or idea that we have of how pathogens are emerging. And I want you to pay particular attention to the timeline here of what we think can happen in really short time frames. Again, this is a model. It's a way to think about this. Very few pathogens have been discovered at this stage of spillover. We don't do enough surveillance to actually be able to track things here. We care about pathogens a little bit more when we start seeing human deaths. But there's a lot happening in the background between, like, from what we think is sort of always happening to what we are seeing as a sort of outbreak, right? And these are examples. I think about 80% of all pathogens roughly have this kind of an origin, where you have a reservoir species or you have a species in which that pathogen is in some kind of equilibrium, where you always get outbreaks, and then sort of the individuals recover, so they have immunity, sometimes lifelong immunity to that pathogen, and these are the kind like bats, some primates, and so on. And then the idea is that when they are in contact with domestic animals and so on, and as you like decrease that space between wild animals and domestic animals and human beings, you have more chances of these kind of interactions happening. And in all of these, we also believe that the pathogen itself will now need to evolve, change, acquire new properties, right? And so having enough number of these instances is going to increase the chance of things happening. And so these are examples of some of the viruses that have emerged with this kind of a pathway. We now live in a very connected world. All of this is stuff that you already know. These are like some of the air routes that connect us. These are some of the shipping routes that connect us. So really anything that appears in any part of the world is going to get around to every other place. And we know now roughly in a couple of weeks. Like if it's a respiratory pathogen, if it's something that spreads really well, we have an understanding of how fast this is. I think you've already seen some example of this, right? Like this kind of hotspot. Why do we have hotspots of disease emergence? Can I ask this question? Okay, so what do you think are the factors that give rise to this? Again, is also a sort of predicted hotspot. So based on factors that we think influence disease emergence, we have these sort of high risk in red and low risk in green. And what do you think is contributing to this?
Yeah, you guys have seen this, so you should be able to answer this in your sleep. Now, come on.
Not to put you on a spot, but just so that, you know.
Probably, as far as I have understood that whatever the countries or whatever the biodiversities that are near to the equator and the temperate regions are usually, they have very considerably high diversity when you consider the density. With respect to density, it has got a higher biodiversity. So those regions are the way density of populations and communities.
Which humans
or humans?
Yeah, humans as well as wildlife, both.
Okay, so high biodiversity
as well as susceptible host and susceptible, potential pathogens.
Let's not talk about susceptible.
Yeah, that's
high human density.
Yes.
And high biodiversity.
Yes.
Where? Temperate or equator? You use both words.
I mean, towards the equator, much more precise.
Great. Anyone else want to add? Land use what?
Changes in land use.
Change in land use, yeah. Changes in land use. Like habitat fragmentation, habitat loss.
What else? Temperature is an important, because it directly also relates to what temperatures pathogens like. Because they are sort of, pathogens are sort of evolving to their host. Our temperature zones are good for them, okay? Above 4, below 60, I think.
I was thinking about health systems. The global north probably has better surveillance systems in place.
But emergence, we're talking about just the potential of emergence here. Here, they are looking at hotspots for emergence, not necessarily where outbreaks will happen or outbreaks will go undetected before they reach some kind of breaking point. So health systems do come into play, but probably at a later stage. And nowhere in the world are there health systems in this kind of space where there is a spillover probability.
One more factor like high livestock, matlab domestic animals.
Correct. Yeah, so basically all of this is going to come back to this, right? All your risk factors are going to come from, are these spaces, are we increasing the probability of this and this, right? And of course, some classes of microorganisms will be particularly good at this. Yeah, sorry, hi.
Hello. So when we talk about the hotspot for pathogens, shouldn't we divide by bacteria and virus? Because the temperatures and the conditions virus-like and bacteria-like are different. So viral diseases are more prone towards the temperate areas, and bacterial diseases are more prone to the tropics. So the showing India and the tropical countries in the red zone probably shows the bacterial and maybe fungal, but not so much for the viral. So there should be a division, don't you think so?
No, like it depends on the kinds of viruses, and also, right? Like for coronaviruses.
This thing I'm saying because I work with the doctors. So what I'm talking about respiratory disorders. What they find is from the patients they are getting. So according to the doctors, mostly bacterial patients are more common in Indian perspective than the viral. I'm not talking about COVID and which came, but emerging in terms of new bacterial diseases come in India versus viral diseases are in temperate.
Sure, I mean, yes, and I'll show you some examples also of what, for example, what you'll see in Uttar Pradesh is different from what you'll find in Assam, is different from what and it's also going to vary by seasons. However, I don't think there is a bacteria to virus divide, and one thing that we must consider with viruses is that you have a really short window in which you can detect viruses. And I'll come to that. When I talk about acute encephalitis, that's one of our major challenges, that the pathogen often disappears before the pathology, like before the person comes to hospital.
Yeah, but just also to say that, there's a similar kind of prediction for filoviruses, for example, potential emergence, and it has a very similar distribution map. So even if you take a very specific, family, just because, biodiversity itself overwhelms a lot of things.
And a big part of the emergence of zoonotic pathogens is also onward transmission from human to human as well. And viruses by far outweigh human to human transmission over any bacterial pathogen, right? Thanks.
Right, so yeah, so I also wanted you to see that there are some temperate regions also. It's not, right? So anyway, so have you spoken about the Koch's postulates before? Okay, brilliant. This.
But we didn't, it's very good.
Okay, so I want to distinguish a little bit between viral discovery versus pathogen discovery, right? So if you take 1 ml of water from the ocean and you try to look for viruses in it, there are really large number of viruses. And if you sequence that and you get a metagenome, that is when you sequence everything in that sample and you try to match it to the databases, which is usually the pathway that we'll follow, and I'll describe more, you will get a large diversity of organisms that haven't been seen before. So you will get them all sitting a little bit on sort of higher parts. So they'll be closer to the root. Have you discussed phylogeny as well a little bit? So they'll be sitting closer to the root. So they will look like they are ancestors. You will not be able to find the leaves because there isn't anything in the database that is close to them. And this happens a lot in marine metagenomes. And I had this idea of showing you that, but I never got around to doing it. But there are these, I'll tell you where to find this data and how you can do this for yourself. And there we are not looking for pathogens. And there, a large number, we expect to see a large amount of diversity. We do see a lot of diversity. There is a lot of unknown. But here, when I'm going to talk about pathogen discovery, we care a lot about association with that particular disease or with what the person is presenting with. And the good example of how to think about it, this was done by Robert Koch, really long ago, okay, maybe 50, 60, 70-ish years ago. And so this is for, I think the first bacteria that was shown to be causal in this way was Bacillus anthracis for anthrax. And what he's sort of, is that right? Does somebody want to say something about that? No, I think that is right. Okay, so the suspected pathogen organism should be present in all cases of the disease and absent from healthy animals. Okay, so basically someone or an animal presents with a particular disease, you get only that, the pathogen that you discover must be present only in the diseased and not in healthy. Okay, so for example, where do you think that this gets broken? Does anybody know? Carriers, asymptomatic infection, all of those cases, this will not be fulfilled. However, this is one of the Koch's postulates. The second is the suspected organism should be grown in pure culture. Okay, where do you think this gets broken? Viruses, if we don't know how to culture them. Fungi, bacteria, if we don't have the right media to culture them. Sometimes the culture times may be really long. We may just not know what are the right conditions in which that organism will grow and simulate it well enough to grow it in culture. But what does this allow you to do? It allows you to say that this particular organism was present in high abundance with some confidence in the person, right? Suppose you don't have it and you're not able to grow it, if you're able to grow, say, multiple organisms, or you detect multiple organisms, it becomes a challenge in actually assigning causality. And this again we will talk about, and I'll try to give you an example where we can only talk about associations from genomic data because we're not able to do some of these things. The cells from a pure culture should cause the disease. So you should be able to inoculate an animal and get back the disease phenotype. Where do you think this breaks down? If you don't have a model organism, it would be quite unethical to grow something and put it into human beings, or don't have a model organisms, don't have a good system. There are ways to circumvent this. Obviously, people are trying to make cell culture methods and so on, but this remains a problem. Then this is the fourth postulate, which is that from this second animal that you have inoculated, you must be able to regrow, re-isolate, and this should be the same as this, the first organism that you isolated. Then only you can assign causality. So this is how it roughly looks. In the case of, so how does this differ from when we do sequencing, find a virus, and we want to ask the question, is this causal? How do you think we can go about saying whether it's causal or not, keeping in mind this is how stringent the criteria are for causality? What are the things that we can do? I'll explain a little bit of this later. So for example, if we change this to sequences, right? We would say the sequences of that organism should be present only in the diseased animal and should not be present in the healthy animal. This seems okay, can be done. The suspected organism should be grown in pure culture, no, but we could say we should be able to isolate the entire genome of that organism. If you put that genome into cell culture or in some other animals, it should be able to give you some some kind of effect, like either the cells should die or something should happen to them if you've got the right system working. You cannot expect the genomic sequences.
We cannot expect the entire genomic sequences to be extracted at any place. For example, if the disease or the symptoms are caused by some kind of toxins or something.
Yeah, here we are only talking about infectious diseases. We are not talking about ones that, right, when you're talking about pathogen discovery.
Thanks, Ansil. I like the idea of using NGS to fill some of these gaps as well, but what do you do when it comes to a pathogen like SARS-CoV-2, where you've got this wide variety of symptoms? You span the whole range of asymptomatic, mild, moderate, severe disease, then point one sort of falls through, no?
Yeah, so actually all of these fall through quite a lot when we look at modern diseases, and no one uses these postulates anymore. But the gap between these and so for SARS-CoV-2, for example, the fact that it is the bona fide pathogen that is causing these respiratory symptoms came much later. what you had was an association of these genomic sequences with the people who had the disease, and then you work your way from there, and then you try to see whether you have evidence for these. And there are other ways to look at causality also. Like for example, the way it was done for finding the association between cigarette smoking and lung cancer, there those kind of causation are also, they are called the Hill's criteria of causation. So then you have some 9 or 10 checklist items that it must fulfill in order. But what you're trying to do is make an argument and then seeing how much evidence is for and against, okay? So this is the concept that I wanted to introduce, that you have to weigh the evidence, and it is not always clear that something that you detected by any kind of testing is the pathogen causing the disease. That usually takes much more work. And for viruses, one of the things that we look for is evidence for exposure. So do you have antibodies or some evidence that you were exposed to this virus? And I'll give you example cases for some of that. This doesn't matter. These are all the different virus discoveries that have happened. And you'll see a lot of these are from marine metagenomes. And then this sort of, I would say that about 2014-15, people felt they had done enough of this, okay? So, so you have, I'll talk about the particular human ones, but I just wanted to tell you that as soon as sequencing became popular and cheap enough, a lot of people were doing metagenomics to figure out, and then sort of figuring out that a lot of things were not present in the databases and so on. So does that, yeah, we'll come back to this, but there's a big list of those. These are lists of actual human pathogens that were, or associated with human beings. I think there's one primate one as well. Yeah, so these were pathogen discoveries that happened. This is, again, a huge list. And this is the sort of way it was done, okay? So there'll be, sometimes you will have culture together with NGS, and then it's telling you what approach was taken. And we'll talk a little bit about this in the afternoon today. That's de novo genome assembly and so on. And this is the disease. And you will now start seeing that in many of these papers, they will talk about strength of association rather than calling this the causal organism, right? So, and I'll give you an example. I think I'm going to give you an example of this particular virus called the Bas Congo virus, which is a rhabdovirus. This is not usually associated with hemorrhagic fever, this particular family, but I think Chandipura and rabies are in this family, right? They're both rhabdos. Okay, so yeah, so this was discovered in 2012, and I'll tell you something about that later. So this basically was one child who died in hospital with hemorrhagic fever, and in a region that did not have Ebola or any of the other hemorrhagic fevers. That region was not known to have other hemorrhagic fevers. So they took the serum of that person. Then subsequently, there was another child who was also ill. And one of the people who took care of them also fell ill. And I forget who they took the serum from to get the virus, but one of them had this single sequence. So I will explain what a sequence is in a little while, but basically think of it as a string of nucleotides, but a really small one. And I think they would have sequenced like millions of reads to get even this single. So I think initially they would have done sequencing, and they detected one small, really small fragment, okay? If you got this kind of data, most people will not have the resources to do the rest of this experiment. So you will have this data and you'll say, oh, I think I have something of the rhabdovirus family, but it may or may not be relevant to this disease because that virus family is not known to cause hemorrhagic fevers. Very hard to make an association in this case. Now, what they did after that is they sequenced a lot. So they took that sample, they sequenced, that means they generated a lot more data. And once they generated a lot more data from it, they were able to get larger pieces. And then they stitched together these pieces in a process that's called de novo assembly, because they didn't think that there was a template that they could map against, okay? And after that, they got this genome here, which is about 12 kb long, and it has a lot of, well, 25% similarity, not a lot of, to rabies virus, okay? And So these are the 3 patients, and they, this is the place and so on. So there were 2 people, we don't know it, from what they did epidemiological like tracing and so on, they never found any contact between these 2 people. This is the nurse who took care of them. And yeah, so basically, and then they took that sequence and they put it on the phylogenetic tree, and it sort of sits separately from all the other rhabdoviruses, but it definitely is a rhabdovirus. And sorry, in that paper then they also looked at, is there evidence for exposure? Is the vector for this a possible vector for this kind of virus present in that region, and so on. But after that, so then they found that there is some exposure, that the population seems to have some antibodies to this virus, but this virus has never been seen again, okay, as far as I know. And so we don't know whether this was really the virus that caused this particular case, or what's happened, or whether it will emerge again, and so on. No, I think sandflies are one of those. It's a vector-borne. I mean, they expect it to be vector-borne based on its family and where it sits and so on. Yeah.
This is really old. I mean,
Yeah.
in the this one of NCBS and
Yeah. This 2012.
in terms of NGS and its evolution, this is a really probably landmark in that sense.
Yeah, for sure. So the reason why I wanted to show you this is because at this time, this had not been done. This kind of work had not been done at all. And it's an important study, but it's also like, now that we are far away from this, we can also ask the question, okay, so how useful was it? It could have been that this could have been an important Ebola-like virus that then spread a lot, and then we would have been more interested in this study. But as it stands, this virus did not come up again as of in the last 12 years. Right? And but the thing is, what it empowers us to do is that if it does come up, we have the primers ready, we have the genome already sort of, looked at in a deep way, we have some assays ready for looking at exposure and so on. And so that sort of goes towards preparedness. All right, so this is just to tell you that as like the cost of sequencing has gone down, we have more sequences coming up. And basically, I'll tell you about the evolution of sequencing technologies as well, and I'll tell you where we are now with nanopore sequencing, which is what I use. Before SARS-CoV-2, right? This gives you an idea. As technology moved, you saw lots of viral sequences come up in the databases. And I should say that around HIV is when we should start seeing. Okay, so HIV was identified somewhere here. Pretty much nothing for the next 10 years or so, and after that you start seeing. But H1N1, that particular pandemic is when you saw a lot of sequences coming up. And why did that happen? Most likely because the technology that you see here was sort of getting ready at that time for doing massively parallel sequencing. Also, it got much, much cheaper. Human Genome Project was the thing that was driving this cost to come down. So things somewhere else affect other things quite a lot, right? So, so now for SARS-CoV-2, I should not put this up. I should have asked you, given that there are about 900,000 sequences of H1N1 from the beginning to now, how many sequences of SARS-CoV-2 would we expect to see? We have had a disproportionate number of SARS-CoV-2 genome sequence. We have 16 million in the GISAID database. Okay, so this is a huge number. And can anyone guess the order of how many sequences we have in India of dengue, Japanese encephalitis, and so on? This is the global order.
All of them together.
All of them together, from 1947, forever. Whole genomes and partial genomes. I don't know if I have that slide, but I do know these numbers.
200,000 going once, 200,000, 200,000.
200,000?
And 200, 200, okay. 10. You're supposed to go higher, but anyway.
I mean,
Anyone else?
You're supposed to use this data to infer that.
Yeah. 1,000?
Okay, so you can see the conservative
No, this is
estimate.
without considering SARS-CoV-2.
Yeah, SARS-CoV-2, I've already given you the
Yeah,
number.
100. So
I'm talking about dengue,
SARS-CoV.
JE, these kind of viruses, roughly.
Complete genomes or?
Okay, you don't, partial genomes, but say greater than 50%.
Then I don't know, it's
Yeah, so
47, 647, very specific number.
6 lakhs.
6 lakhs. Wow, 6 into 10 to the 5. You said 10?
altogether also, yeah. So basically, it's a few hundred. I think 300 for dengue, maybe less than 100 for JE. Just enough so that we can make some diagnostic and something, right? That's how much sequencing we were doing. So for us to scale to this, it was quite something. But in the last year, we started seeing more of the others as well, like chikungunya and dengue and so on, but still not approaching even the tens of thousands. And the reason why it matters is many of these are RNA viruses. They are changing quite a lot, and it is good to track how they are changing. So, okay, so this is possible, but I don't even think that we are sequencing. Even in like what people are not putting up in like databases, I don't think it would cross more than thousands for each of these pathogens. Because earlier what people were doing, because the technique that people were using before is they wanted to do culture followed by sequencing. Now that's very cumbersome, and you need to have the right lab facilities and so on to do that. This idea of doing amplicon sequencing directly from the sample and so on is new for us. And so it hasn't been done enough. And it definitely was not a thing that was done before SARS-CoV-2.
Sorry, Chitra, I might be digressing a bit, but is there an agency in India that's mandated with sequencing emerging pathogens? Like in Canada, we've got the Public Health Agency of Canada that does that for us.
So the NIV, National Institute of Virology, has all the specimens, and they can do, and they have been pushing this the most. There are also these labs that are regional labs called VRDLs, which are supposed to be this virus research labs across the country, which will also then contribute to the sequencing effort. There's also this national consortium which did the SARS-CoV-2 sequencing called INSACOG, which is actually a combination of a lot of research labs. They had the sequencing capacity, but not the right sample. So then you have to match those two, right? So unless you're working on a pathogen, you are not going towards sequencing it in any way. And then if you wanted to sequence it, you didn't have the facility to do it and so on. So when we're talking about pathogen discovery of unknown things, it matters because the facility to do this and the ability to do this actually in very small pockets in the country. That's sort of what I was alluding to in this whole thing. Right?
And
Yes.
just to point out that actually Chitra has been part of that history, really pushing that forward.
Thank you.
So does this all a part of IDSP?
So IDSP reports, so this is an integrated disease surveillance program. They are looking at things from a public health perspective. They report outbreaks of disease, but they are doing sort of symptomatic or syndromic, that's what they call it. They call it syndromic surveillance, which means I will report something as brain infection or acute encephalitis, but I don't have to tell you the cause. And I definitely don't have to do the molecular diagnostics for it. But the hope is that the IDSP network will talk to this VRDL network, and together they will bridge this gap. It's taking some time, but it's meant to happen, right? And there's also this, so IDSP comes under this organization called NCDC. You will be hearing more about them and on the outbreak response. So yeah, so this is what we'll be talking about. Identifying a novel sequence as a potential pathogen, establishing association with disease and obtaining the complete genome, addressing causality. So usually for addressing causality, you then need orthogonal approaches. That is, you can't tell it by the sequence alone. You have to look at immune exposure. You have to look for possible vectors, whether the vectors are present, etc. So you have to make a case, build up for that. Outbreak investigation, you will also be hearing more about this in this course, but usually good to have a cluster of cases for outbreak investigations. And I'm going to tell you a little bit, I think, around this. Yeah, and serology gives you an idea of exposure history. So basically, serology is looking at whether you have antibodies towards to something. And one of the criteria in the Hill's criteria for causation is that if you think something is causing the disease, if you treat it, the disease should not be there. Okay, so yeah, so if you want to look at the Hill's criteria for causation more, this is a good link. So what do you do when a person, or when you get a cluster of cases that have no known etiology, right? The first thing as a microbiologist, what you would like to do is take the specimens from the person or animal and look under a microscope. Now, what do you think needs to be there for something to work, for you to be able to see things in a microscope? Okay, so there is a limit of for light microscopy, but say I'm able to do electron microscopy and everything. What is still a limitation of microscopy?
I mean,
You have to have the right sample. You can biopsy a dead animal or dead person. Huh. So you have to, yeah, so the pathogen must be present in the sample that you're looking at. It must be in the range for that kind of microscopy, in size range. And what do you think is the third thing? And this is an important thing, that's why I'm pushing for this one. What is the biggest challenge? Does anybody know about the diagnosis of TB? What is the biggest challenge when you're trying to diagnose TB? Not enough of that pathogen, not enough is in that sample that you're testing. And in general, there might be low load of the pathogen, right? And so microscopy usually requires a lot of the pathogen to be present for you to be able to see it. Growing it, what are the challenges? We already discussed. You need to know how to grow it. But traditionally, microbiology has relied on these two, right? You grow a pathogen and then you do everything else. In fact, even today in clinical microbiology, if you cannot grow a pathogen, making a case for it is actually quite hard. Okay, and people will be very cautious about that interpretation. Only now we see a change towards molecular diagnostics. PCR, people are willing to accept that a bit more now. Is everyone familiar with what PCR is?
Yeah, everybody knows what PCR is, right?
Yeah, we'll talk about it a little bit, but this polymerase chain reaction, it basically is a way of detecting, once you know something, it's a way of detecting that particular organism or any gene or anything, okay? I'll discuss a little bit more about that. Then of course there are methods that rely on host defense response. So all of these antibodies and so on are the host making a response to the pathogen. So all these, any kind of IgG, IgM, antibody detection technique, you will usually hear about this when you're doing dengue testing. You would have done IgG, IgM, no? That's sort of looking at exposure, right? And IgM usually means acute, that is you got it right now. IgG is past exposure, roughly. A test for known organisms is usually via PCR, or you can have some kind of antigen test. Sequence everything is metagenomics, which is, we don't know what's in the sample, let's sequence everything there and see what's in it. Why do you think this is a problem? Like, why don't we just do this for everything? Just sequence everything, catalog very nicely. It's expensive, okay, but suppose like unlimited resources. It's bad, computationally it's bad, but not too bad, given how much computation we are able to handle today, right? Not too bad. But you can run it in an online thing and then destroy the data or something. Store it in a format that you don't need, like, I suppose that those are challenges. There is one more challenge with it that we will come to. It is not very sensitive, okay? You need to sequence at really high depth. And again, the same problem of microscopy comes up here. If the pathogen is not present in high enough numbers and is still causing disease, then this doesn't improve very much. Like, you will get 1 or 2 sequences. You saw, right, in that one paper that I showed you, there was one small bit. Then they sequenced a lot and got the other. Sometimes you sequence a lot and still don't get the entire genome. Okay, so that paper is a bit of a lucky thing. Direct PCR, the primers have to be right. What does that mean? It depends on you knowing the region that you're looking for well. And if the virus has changed or the pathogen has changed, or for bacteria, if that region is not present in the particular bacteria that you're looking at, then you will not get a PCR result. Multiplex PCR, again, there are sensitivity issues. So basically, if you want to detect multiple things, the number of copies that you can detect them at and so on, you need to have all the primers working, plus you need to have enough of the pathogen. Then there is 16S sequencing. 16S is basically a region of the bacteria that is conserved very well. And you can use that to figure out what are the different bacterial species present in your sample. And so that's a sort of shortcut to doing this full metagenomic approach. It gives you an idea of the bacterial diversity. That is used, and there are some clinical, there are some places where they do use that, but it's again, it gives you a limited amount of information. You can't always come down to which species it is. You get to know which family it is, but you don't always get the final. And many of these families, they are very close. For bacteria, they are like very close to each other. There'll be a non-pathogenic E. coli sitting next to a very pathogenic E. coli, and all you will get is E. coli. Then it'll be a problem. So that's that. Then you have targeted NGS. This is what we have used for sequencing SARS-CoV-2 a lot, many of the other pathogens. When you know a genome, you build tiling primers. That is, you have primers throughout that genome. And then you get that entire genome out. So that's targeted NGS. There are also other ways of doing targeted NGS. One is called WRCAP sequencing, where you enrich for things. So you know there's a virus in your sample. You use small bits of all viruses that are there to pull out from your sample, okay? This is roughly what it is. So you have small bits. So there's a reagent cost there to make that library of small bits of all viruses and bacteria and so on. And then you pull it out from your sample. There's an enrichment method. So, and then you sort of do a PCR, increase the number of things and sequence it. So then there's metagenomic sequencing. There is serology. Serology is looking for the antibodies, microscopy. And there's something called MALDI-TOF that is it's a sort of looking for peptides or known. Once you know something, I have heard that in clinical use, it's actually getting used a lot to identify pathogens because it's very specific, pretty good, easy to do, and so they like it. So if you're able to come to that level as a reagent.
It's a really important table.
Sorry, I will
no,
send it out.
I'm saying it will be great to get it later.
I will send it out. So, right. So now we'll come to...
But just one second. So basically, like, I think what's really important, you can see that technology has really impacted the way we are able to do science. Sometimes we think of science as independent from technology. For example, maybe ICTS, a lot of the science that happens here is independent of technology. But in this case, technology has really impacted our ability to discover. How it has impacted what we infer and how we move forward is something to think about.
Yeah. I think for sequencing and for pathogen, like looking at pathogens by sequencing, it's definitely, it has moved with the technology. And these are the three different kinds of sequencing platforms that are there. I'm sorry about this. Can I hide it? Sorry, making it worse. I'm also on an Ubuntu system. No, I'm not anymore. Okay. I have a feeling I stopped sharing also in between, which would be bad. It is shared. Okay. Hide floating meeting controls. Control Alt Shift H. I'll ask you to remember that. Sorry. Okay, so in the beginning, when sequencing started, okay, does everybody know what DNA is? Think of DNA as this long string. No, I mean, not everyone is from the same background here. So, right, so think of DNA as a string, right? And think of that string being composed of 4 colored beads. What matters is not how many of these beads there are, but what is the sequence or arrangement of them, okay? So, so that arrangement or sequence of those 4 colored beads, they are actually 4 different chemicals, A, T, G, C, and how they appear is going to distinguish all the different molecules that we have. Now, the way we are lucky is that all life forms that we know about have the same basic genetic chemistry, okay, the chemistry for their genome. So any DNA that you take, whether it's a plant, bacteria, virus, it's going to differ only in the backbone sugar, that is RNA or DNA, but not in these 4 chemicals, that is ATGC, that makes them up. Okay, instead of T, there is a U for RNA, but in principle, the idea is the same, that the way these chemicals are arranged is what determines what kind of properties you will get. because the DNA codes for proteins, and these proteins are responsible for what happens. Most of your functional things happen through proteins. So what we are trying to understand when we are doing sequencing is looking at the arrangement of these chemicals. So all of these sequencing technologies are trying to read ATGC and which position they come in. When we talk about errors of sequencing, they are saying that actually we can't be sure about whether it was an A, T, G, or C. Okay, so that is what the errors of sequencing look like. In the first generation of sequencing, what you could sequence was small fragments, like 500 to 1,000 base pairs. So the length of that string is about 500 to 1,000 base pairs. Roughly, to give you an idea of how big is the human genome, does anybody know? 1 billion such base pairs, okay, 3.6. 3.6 into 10 power 9, okay, base pairs. How big is the SARS-CoV-2 genome? 29, so about, yeah, 30,000, 20, yeah. And dengue? Yeah, yeah, good. So the length of these strings vary. And so some of the sequencing technologies will cover it faster than others, right? So if you are able to cover 500 base pairs or 1,000 base pairs, roughly 10 reads that are sort of next to each other will cover the genome. All right, for dengue. Now, for the second generation of technology, it was massive. So these actually, you could only sequence a single such string, okay? And this was a problem with this technology. It was very low throughput. Then the technology became such that there was high throughput sequencing. This is when you start seeing a lot of the sequencing data also coming into the databases. So you get massive parallel sequencing, and I'll show you if possible how, and you get between 50 to 500 base pairs there. And now the technology that we have can do both this parallel as well as single molecules. So there is a difference in this third generation technology, and it also can do lots of long read sequencing. Yeah, not with these. You require different. Yeah.
Instruments are completely different.
Yes. Yes. Yeah. Because the synthesis is more like chemical reaction. Yeah. The sequencers are not the same as the synthesizers.
Synthesizers. Okay.
Yeah.
Get the mic. Yeah.
Okay. I just asked if we can do synthesis and sequencing in the same instrument or not. That's it, ma'am.
That's a nice idea, if someone is able to do that. Okay. Again, these are the kinds of technologies and kind of companies that make them, and then sort of read lengths and output and how much data they generate, and that becomes an important thing. Like how much data do you generate in order to then be able to do the analysis to look for new pathogens. So this is a better explanation. So for the second generation of sequencing technologies, what happens is you have the genome, that is the DNA, you cut it into small pieces, either enzymatically or by sonication or something like that. Then you add, and this is, I want you to pay attention to this, because what you do is you add known sequences to the ends of your DNA. And so when you're doing your bioinformatic analysis, you will get a lot of these sequences also, okay? And that's something to keep in mind when you're doing that kind of analysis. And then what happens is you get these DNA to be going and sitting on a particular, so you can get 3 different kinds of amplification. These are 3 technologies. Here is sequencing by ligation, where there's a DNA ligase that sits and does the sequencing. As far as I know, this is not being used too much anymore. This is pyrosequencing, which looks at an ATP being released when the DNA is getting synthesized. And then in this, there is a proton that is released. So for DNA, what happens is there's one strand that's present, and this strand is a complementary strand. For every A in the first strand, there's a T in the second strand. For every G, there's a C, and vice versa. So that's how all the sequencing by synthesis works. So in this case, the proton that's getting detected, in the case of Illumina, I think, you get a fluorescence being released. And There is, if you want to check it out, there's a YouTube link here. It does a really good job of explaining the entire sequencing process for Illumina. And this is how the data looks like, okay? This is what the camera is capturing. So there are many different clusters, and each of them has a particular fluorescence signal. At that time, it means a particular DNTP. That is, either ATGC is what you will see from that cluster. So each of these is going to generate a sequence. Then at a different point in time, all of these colors are going to change depending on what's present in that particular strand and so on. So this is roughly, it's just for you to know where the data is coming from. In the case of nanopore, what you have is a membrane and you have these biological pores embedded on it. The DNA sits on top of it because of this helicase that likes to sit on top of this particular protein, and it unravels this DNA into a single strand. So the DNA goes through it, and as it's going through it, you're getting electrical signals from this part of the pore, which is usually a 5-mer or a 6-mer. That is, the length of the string is that much, and that's giving you an electrical signal here. These are called squiggles, and these need to be converted into base pairs. So you need to infer what is the chemical composition of these 5 things here that is giving rise to this signal. And that's why you have in nanopore a step called base calling. And that step actually improves as this pattern recognition algorithm improves. So with nanopore, you can have the same data, but then get slightly different results as the technology improves. This is how the machine looks for nanopore. This is their newest, one of their newer machines, where there is a computer along with a sequencer inbuilt. It's this big, okay? And compared to Illumina and other machines, which are quite large and you need AC and dust freeness and so on, nanopore doesn't need any of that. It has other problems, okay? Just ask me about the problems and I will tell you about those. But so this part here that you see is a flow cell, and all the membrane and things that I told you about, they sit here in this place. About 2,000 of the pores are here. And all of this is the microfluidics to clean it and buffer it and keep the biological proteins happy and so on. And bubbles are the enemy of nanopore. So any bubble, air bubble that gets into the system, no sequencing happens. Okay, so this is a sequencing going on, and this is just, again, basically how we prepare. So this thing, what you put inside a machine, that's called a sequencing library. So you will hear this term often. It's basically nothing but your DNA prepared in a certain way with adapters added to it so that the machine can recognize it and put into the machine for sequencing. Why am I using this? Okay, so when you look at this kind of data that comes out, we want to ask the kind of question that you should be asking when you see this kind of data is, do we know that the experiment that was done before even interpreting anything about a pathogen or not there, present, not present? These are the things you should be looking at. First is accuracy. You look at the wet lab protocol. Can that protocol detect known samples? So if I have a flu sample or a strep pneumonia sample and I put it in, am I able to detect it by my method? So you should have known samples confirmed or detected using other molecular techniques and see whether it can be done, okay? So you will know roughly whether you are able to detect that kind of pathogen and your protocol is working. These are not easy protocols to get them to work in every circumstance, okay? They tend to be very finicky. They are mostly in research use. There are only 2 labs in the world that use them in diagnostics. One is in UCSF and the other is in London. Yeah, so the dry lab, the next thing that you need to do is when you get these sequences, and we're going to talk about the bioinformatics in the next session, you'll be comparing them with things in databases, okay? And if your database is bad, or someone, for example, there is something in the database called arena virus, okay? It exists in the NCBI database. It's actually something else together with an arena virus. However, this part of the sequence is actually an adapter. But now, every time I use my algorithms to look for a match, it will match an arena virus, but actually it's matching the adapter. So those kind of problems exist, so people are trying to standardize the reference databases for microbial sequences.
We are talking about several technologies and stuff here. So from a disease point of view, like you gave the example of, I think it was BASV infection and say dengue also, right? Deciding what methodology to use to detect an infection, I think it's a very big problem. Like even today, so I want you to kind of dwell on that a bit. say, deciding would you do sequencing for dengue because a patient might be infected with, say, 2-3 different kind of things, right, going on at the same, like in our country. Serology versus PCR versus sequencing, what would be the advantage of?
Yeah, so I mean, yeah, sure. So basically, ideally, you are going to go with what's available near the person first, right? So most of these tests are available in research labs or tertiary care. So it's more about access as well. Most places are not going to have sequencing, and you're going to be doing sequencing as part of a research study. Even the labs that do use sequencing for routine diagnostics do it in very specific cases. For example, in acute encephalitis, it has been used in brain infections because the 48-hour window, I think, is good to know how to treat the person. So it depends a little bit. I'm not answering your question fully, but it depends a little bit on access. And it is also the clinical suspicion. If you expect that the window for the virus being there has already passed, for example, let's look at dengue, right? Dengue, I think you get infected, then you have an antigen, that is, you have the virus producing an antigen called NS1. Now that's present between 1 to 3 days or 1 to 5 days, peaking at about 3 days. If you're detecting it after 5 days, you will not detect that because it's not in your blood anymore. So then it's not the virus, but what the virus has triggered that you will look for. So then you will look for IgM, which sort of starts at 5 days and stays for about 2 weeks or something, but we don't know when it fully degrades. So now you'll be looking for IgM. If you're looking after 2 weeks, you'll be looking for the IgG or the antibody that stays after that. So when you, suppose you're doing a PCR, which is the right window to do a PCR? The right window is when the virus is increasing, that is the first 5 days. So you will often hear people write this statement that acute febrile illness, first 5 days post fever or something like that. That's because we don't expect, although there will be a lot of variation for an individual, right? At a population level, this is what our expectation is, that within the first 5 days, you will not see virus in the blood anymore, so you shouldn't look. So looking for viral antigens is actually a very short window that you get to look for them. And we have to be careful about that because all of these techniques, many of these are looking for that, which is a short-lived thing, right? And so if you're looking at a population level, then you have a better, so if you're pooling samples from many different people, that's why I said cluster of cases a bit better. So if you have a cluster of cases and you have tried to do this, then you have a better chance. So you're increasing the probability of detecting something. I'll come back to it. Okay, so then, I mean, this is how to do a good metagenomic experiment. Now there are some papers and there's also some guidelines around what you should be doing. What you get at the end of it is sequences belonging to many different organisms. This is a kind of plot called Krona plot. You should familiarize yourself with it because it's used quite a lot in this field. Because what you rarely have a sample that's completely blank, okay? The problem is that you get a laundry list of agents from a sample. Usually this can, because this, while it's not, so if it'll be like I use water in my sequencing experiment, I use a lot of other things, I'm gonna get all of those things in my sample no matter what. So you will have a background and you'll have to separate the background from the signal. Our usual workflow is you take the sequencing data and you remove the host sequences. I have some more stuff to share around this, but I'm wondering whether we should take a break or when is... 10:30 is the break. Okay, cool. We can, yeah, we can have more discussions. I was just thinking what I'm gonna... Okay, so after this, what I'm going to take you through is one example of a cluster of cases, and then one example of brain infections. And the closest we have come to pathogen discovery, which is not, it's not pathogen discovery, it's just pathogen identification, but that's where we are right now for the reasons that I have told you already. Yes.
So we have a few diseases rapid kits. So does it always have to be once the disease is confirmed with the rapid kit? Should we go for something like molecular diagnostics? Is it always necessary, or it's fine to rely only on the rapid kit in some cases?
So it depends on the sensitivity and specificity of your rapid kit.
Right.
The reason why they usually ask for a confirmatory test after a rapid test is if either the sensitivity or specificity are not very good. So if they're 60% or something like that, then you want to confirm. That's the thing you have to look at. There's no general rule that I know. Yeah, I'm reasonably sure. Yes.
There is one more doubt. We have already enormous amount of complete genome sequences of so many different organisms. How do I know which one is the right one? I mean, as you told that adapters are always the problem. So is there any way to find that whether that sequence has got any adapters that were used for the sequencing?
Unfortunately not, and that is why people are building curated databases. So you'll find companies that are doing this kind of work, and what they are doing is doing the work of curating these databases. Because what needs to happen for something like this to get removed from the NCBI database is that the people who put the sequence in have to submit a revision, and it's very hard to get someone else to do that.
For example, in case of dengue, so if we see there are so many sequences, and especially the variation comes in the untranslated regions of this. So if you see some 5' untranslated regions are very long for some strains, and for some shorter for some strains. So is there any chance that the adapters might be causing this variation, or
Yeah, for dengue, I'll tell you why this happens. It's also, of course, the various methods of sequencing can introduce lots of artifacts, right? There is, I think, also a circular stage in the flavivirus's replication. So you can... So it depends on how the person has done the bioinformatics. Sometimes we can get larger sequences, then you need to do the clipping yourself. Curation of sequences by an external agency is rarely done right now. Curated databases usually are proprietary and belong to companies. They do have them, but...
Yeah, this is a very important issue about the database itself. For example, with coronaviruses, a lot of people label sequences based on the host. And so, for example, if you're trying to build a phylogeny or something, you want actually kind of closely related or taxa of coronaviruses, but they're all labeled by host. So it becomes really difficult with pathogens because we know so little, right?
Yeah.
With viruses.
Yeah, so for viruses, there is this committee called ICTV that sits and tries to do some kind of normalization of these kind of things and try to get place names out of it and have some kind of systematic way of labeling things. And there are for pathogens, I mean, like I told you, this Argos or whatever, they are trying to do this sort of systematic looking at, but they want to, I think their approach is from the beginning, we'll control everything. So we'll make sure that this pathogen is sequenced twice, 2 different platforms, gives the same results. So it's very, very stringent. It's taking it looks like it is slow. Looking at the amount of data that is there compared to NCBI or nucleotide database or SRA or any of these, which are very rich in the amount of data, but the quality you have. So basically you have to go back and check some of the things that you're doing and align, and in this case clip and so on. And adapters, you will have to basically do your own checks. Yeah. Any other questions?
I was just curious to know, like, while we give samples for sequencing, so in case if there are any errors, or how would you interpret it?
What kind of errors? Like what stage?
I mean, after you give your sample in the first stage or the second, any stage when you...
So basically you have a sample, you extracted the DNA and you gave it to DNA only, or you made the library yourself?
DNA.
Okay, so then when you are giving just the nucleic acid, right, then you have to know what technology they used for making the library. That is, how did they chop up that DNA? Did they chop it up at all or not? If it's what process did they use? Did they convert the RNA that was there into DNA? For example, if you're looking for an RNA virus, you didn't do this step of reverse transcribing it, then you won't get it, right? So you have to know all the details, and then if the library is not good, there are some quality parameters for a library. So after PCR, you expect a certain yield. If you don't get that yield, then you know that something went wrong. Then again, the adapter sequences that are attached, what is the efficacy with which the enzyme is able to do that? So you might have some loose sequences that do not have an adapter attached to it, which will then contaminate your reaction. If your DNA is degraded or is not of high quality or has some other things then you may not get a good library prep. So there are a lot of quality controls during the library prep before something is sent into a sequencer. Once you have, so that is when you will get a quality report back. Usually it will be on the parameters of like nucleic acid purity and length of the fragments that they were able to get. Suppose you get really low fragments, everything is like 25 base pairs, most likely adapters, nothing else got enriched. So those kind of things. And when you get your data back, then you can do some QCs, and I'm going to show you some of those QC steps in the next session.
Sure. Thank you.
Okay, yes.
Yeah, so when do you use Illumina and when do you use nanopore?
Okay. Okay. It's a little bit of Illumina is a technology that's earlier than the nanopore. Okay, nanopore is a black box technology. They don't usually tell you what's. So basically, you get a consumable that's very, very sensitive. Illumina also, you get consumables that are very sensitive, but there are sequencing labs. So they are doing this day in and day out. The process is the same. Nanopore, you are trying to do some of this yourself for the first time. So it's, at least I would say 5 years back, I was calling nanopore the bleeding edge. But you can take the nanopore anywhere in the world. So I'll show you an example where we've taken it to Uttar Pradesh in the middle of an outbreak and sequenced it there. You cannot do that with Illumina. You'll have to buy that machine there. You need power backups and so on. Nanopore I can run from my laptop. So, you know, it depends on those kind of things. Then there is the error rate. So the reason why I said that the nanopore data improves with time as the pattern recognition improves is because the error rate of nanopore is actually very high. Okay, and it is taking a single molecule and it is sequencing it. Illumina, very good as far as error is concerned. So if you are looking for mutations and single nucleotide polymorphisms, much better, much easier, more standardized protocols to do it with Illumina. If you're trying to do that with nanopore, you have to put a lot of checks in place and check things manually. So when we have reported SARS-CoV-2 variants and done it by a nanopore, we have manually checked the data to see if there's a possibility that got misaligned, something happened, etc., etc. Nowadays, there are better ways to do error correction as well. However, I have heard that the new reagents of nanopore are better and are giving higher quality. Again, there is a thing called Q-score. And I'll tell you a little bit about that as well. Nanopore tends to be bad, Illumina tends to be really good. Nanopore is improving. Illumina is, what's happening with Illumina? Illumina is becoming portable. They are having some smaller sequencers as well now. So,
ma'am, one last question. What is the cost of this nanopore instrument? And what is the per base pair cost?
Yeah, so I had that somewhere, didn't I? Okay, I don't have the cost here. Okay, no, it's hard to do that kind of comparison, but with nanopore, I think the instrument setup cost together with computer and everything will be less than 5 lakhs. However, every single experiment is going to cost you about 1 lakh. Okay, so is that?
To bring up when you're talking about technology and how technology is changing a lot of things. I think in India, we have a lot of barriers to that because basically about the quanta, it's mainly about the quanta of funding. And I might be wrong. I've been faculty at NCBS for 20 years. But, you know, with granting agencies and stuff, when you're talking about tens of thousands, that's one thing. But when every unit of consumable is in a quanta of lakhs, that is something which becomes very, very challenging for Indian researchers. So something we should remember also that, I mean, I'm not solving the problem or anything, but saying that these kind of challenges especially hit researchers in India.
I'm not offering a solution, but maybe potentially a solution. Are there barriers to collaborating with researchers that can bring in international funds?
Heavy barriers. heavy barriers, especially in the context of disease work, because of national primacy or whatever you might want to call it. Sharing of data not allowed. International collaborations literally impossible.
Yeah.
Sorry, maybe that's my experience and maybe it's, Sorry, maybe there's
But
other
you are the
experiences.
most experienced in
But
this.
it's not allowed. There's just barriers. And all collaborations internationally have to be approved by ministries, which means 6
So
to
it's
8
really
months,
slow.
more than a year. Yeah.
It's really slow. So every advantage that you expect to gain out of being ahead of the technology, you will lose that. I mean, there is no way that there's a cutting edge technology that you can use it in India at the same time that it's being used somewhere else. You are a few years behind, and that's very painful, particularly for younger people. But anyway, I don't think there are good solutions to that. Because it also takes, it's more expensive for us to get the reagents. It takes longer for us to get it. And then there are all these access challenges. So nanopore was difficult before, but now they have sort of India team, and they also have manufacturing, I think, in Singapore now. So you're getting things faster, more streamlined, and less problems. Sorry, was there any other question around this? So what you should look at is your per genome costs, right? How much does it cost you to get a genome? And that is usually comparable if you're able to multiplex correctly.
Yeah, but also with something like Illumina, the cost goes down with high throughput. And again, so basically we are in that density dependent, like, alley effect, like we are always finding it difficult because there are fewer people, less money, changes in technology require more playing around and all of those things.
And all of this, of course, you're relying on PhD students and so on. So you have a number of years within which the project must complete. So, yeah. Yeah. Like the number, yeah, the number of incomplete analysis that I have is ridiculous. Okay, so cluster of cases. This is what the report looked like on 5th January 2020. It's a pneumonia of unknown cause. So this fits within what we think can be addressed by pathogen discovery. Okay, so you had people, you didn't know whether the virus was transmitting among people at that time, I think. According to authorities, there were people who were in this seafood market, and based on preliminary information, there was no evidence of significant human-to-human transmission. No healthcare worker infections were reported at that time. 120 close contacts were under medical observation. So something was happening. Cluster of cases were there, respiratory pathogen, good case for doing pathogen discovery. So people went there, they actually did sequencing. They found that the SARS-CoV-2 that they found is somewhere here, right? Yeah. So it actually does not sit with the other SARS-CoV-1. It sits differently in the coronavirus tree, closer to some bat coronaviruses. And this has been filled up a lot, okay, over time. I don't remember that this is the 2019 paper, but after that we know other close relatives as well. But this is just to give you an idea of having all of this background was important to be able to place this on the tree, right? If you did not have this, then it would be difficult to place. What did it immediately give you? It immediately gave you a sense of what the receptor for this particular virus was going to be. So this is 2019, right? You have the sequence that allows you to put it on the phylogenetic tree, tells you it's a SARS-CoV-2, tells you that it is a coronavirus that is different from MERS, that doesn't seem to be like the other one. So you can expect that it's going to be novel. People are not going to have pre-existing immunity towards it. It binds to the ACE2 receptor. So that's a lot to know quite quickly because of all the other work that has been done. This work came out much later, but this is to give you a sense of, because I think it's relevant for this course. That's why I thought I will talk about this a little bit. So this is looking at the Hunanan market and looking at where the cases were distributed, and then some hotspot analysis, which later retrospectively told us that it's probably the market that was the epicenter. And this market was shut down very quickly because you're not supposed to have wet markets with multiple species and so on, because the origin of SARS-CoV-1 was already known to have this kind of origin, okay? So there was a lot of clampdown, so people didn't want to talk about it too much either. They just shut down the market, everything was rinsed, cleaned, whatever. And these were images taken by someone before this market, like a few years before, I think. Somebody had gone and seen all of these different animals being there. And so this is the sort of, this is the map of the market. This is where we think there was some virus circulation in the environmental samples that were taken much later. Like this is, it has been shut down, everything. So basically, I think this is somewhere in January where they went and took this. And they've gone and taken environmental samples in the warehouses, in the sewage, etc. So these are swab samples and so on. And they even isolated virus from that. Okay, so definitely present in that location. Then this is a little bit of a cheeky bit of work that was done. So this part of it, we'll come back to what it told us, but I wanted to introduce this. So the people who put up this data, right, of these sequences from all of these swabs, they also had put up metagenomic sequencing. So some people had this idea that, oh, if we have the metagenomic sequences, we have the DNA of the hosts present in it as well. I sequenced everything from the sample. So then they tried to use mitochondrial DNA to trace what are the different organisms that could have been there, okay? And so they had all these different species that they thought were sold there and other mammals. And then they sort of put that on a phylogenetic tree, market samples together with what was known about these. And they sort of came up with this idea of what were the animals, like this Korean raccoon dog, then bamboo rat and hedgehog, and all of these different animals were probably in proximity with each other and being sold in this. I don't know if they've taken this paper down, but for a short period of time, this was online and available. So what is that idea that gives us, right? The idea that so the idea that researchers have from that is that based on the SARS-CoV-2 phylogeny, they think that actually there were two independent lineages that were circulating in the market. So there was enough of the virus for it to acquire mutations and go through many different kinds of hosts. And we already had lineage A and B. And if for those of you who remember, very early on when we were sequencing, we actually found both lineages. So both actually got out of the market and into the world, is our idea of it as it stands today. And there are some parts of this story that we are doing retrospective analysis, right? So some part of the story we can never confirm. So anyway, so these are the lineages that were found in the market. So the thinking is that there were intermediate hosts there and multiple hosts. Then you had two different viruses getting introduced and surviving in human populations and transmitting onward. So this is the basic idea that there might have been some kind of reservoir, possibly bats, and they were brought to this market from an unknown location and probably in cages in that particular area of the market where you found most of the environmental samples to be positive, and then you had two lineages that subsequently traveled the world. So that's an example of a pathogen discovery. We went quite fast with it because we had other known pathogens close to that pathogen, and that helped move things along further. This is an example of a pathogen discovery or pathogen identification approach. So from now on, we're not going to call it discovery, we're going to call it identifying pathogens and associating them with disease. And here, this is unbiased sequencing, that is metagenomic sequencing, to detect in brain infections particularly. So this is Charles Chiu's group, and what they did was they have shown before that if they use this approach, they can detect pathogens in a 48-hour window. And they have detected pathogens like leptospira. Okay, so this is the history of this group. And now they are saying, hey, but which method is better? Should we be doing Illumina? Should we be doing digital PCR? What helps? Right, so they've done everything in this particular paper, and you can go back and look at it to see, and they got some reasonable result. But one of the critical take-homes of this paper was that you have to sit with the clinical team after you get your bioinformatics results and ask the question, does this fit the clinical picture? Because what you get is a list of pathogens or list of potential pathogens or list of opportunistic infections, and you don't know what to make of it. And they have this method of, they say that you look at how many reads you got or sequences that you got of a particular pathogen in your sample, but you try to normalize it to the background because you know that your background gives you a lot of data. So have a water control and these kind of things and normalize and plot it like this. That's sort of what they say. Brain infections, big challenge. You have encephalitis and meningitis and everything in between. So things that affect the brain itself and the meninges, that is the covering of the brain, and lots of different pathogens can cause brain infections. This is part of the problem. In India, occurs as outbreak in northern India, northeast, endemic to South India, so you get cases throughout the year. And 70% of the cases, we don't know what's causing them. There is some seasonality, particularly in the north and northeast, affects mainly children or used to affect mainly children. And some of it is Japanese encephalitis virus, but what is the rest? So this is the question. So is JE testing bad, or are there other pathogens out there? So this is what I started looking at about roughly 10 years ago. And what we started doing was we took cerebrospinal fluid, we put in some known vaccines actually, and tried to see if we can detect. So I told you one of the things we need to do is we need to show that our method can actually detect pathogens. So we put in the MMR vaccine, showed that we can detect it. What is the limit that we can detect? We can detect 28 particles, 14 particles, and so on, okay, with confidence. That we got large coverage of the genome. So this is the genome, 14,000 base pairs long. We got about 100 reads mapping to every bit of it. That gives you reasonably high confidence that the pathogen is present, okay. Yeah, so we did a little bit of an analysis from an cerebrospinal fluid that had been collected by Dr. Shahid Jameel from an outbreak in North India, and we just did an analysis in that to see what's in it. And with every sample, we didn't get a single organism. We often got multiple organisms in them. And these were some of the examples. So Japanese encephalitis virus, known to cause brain infections, sure, no problem. Cryptococcus neoformans, again, known to cause brain infections, not a problem. So Alternaria alternata, it's a fungus, it's present everywhere. Probably not a pathogen, but this is the only thing we got here, okay? And we also got many viruses of the fungi. However, did we get it from the tip of the pipette that we were using, or the water, or so on? So this experiment taught us that we had to build in a lot of controls. And also these samples had been collected somewhere, stored somewhere, brought. All of those need to be tracked if you want to make any kind of association. We got this virus again and again called aphid lethal paralysis virus, and I was initially very excited about it. Insect viruses, you can't make a good case that they cause anything in humans, okay? Anyway, so these kind of things is what you get. This cardiopyrovirus, we got a very small bit of it, and we've never really gone back and looked at this data carefully to see if there's more. But this belongs to a family where a sister virus of this virus is called Banna virus, this one, and it causes encephalitis. And we got a really small hit. This is how, it was 50 base pairs or something. So again, we didn't, but it was a good hit. That is, you can see that it's matching almost everywhere and doesn't match to anything else in the database. But did it cause encephalitis or not? We don't know. So these are the kind of things that we are worried about. We care about contamination. We care about the skin microbiome because you're handling it. And then when you take the cerebrospinal fluid, you need to puncture through the skin. So, right, so you might get skin commensals. Then distinguishing commensals from genetically related pathogens, opportunistic infections. So these are hard. So why do we get, so at the end of this exercise, in order to understand, this is sort of my understanding of what impacts pathogen discovery type work. One is the quality and the quantity of the sample, limitations of the method itself, that is, we are looking, this was for PCR-based methods, not for NGS, but low pathogen load is a problem for us. And then you have poor databases to compare our sequences. This is a lesser problem now because I'll tell you about the computational approaches we use. Even a little bit of similarity can help us zoom in, but these are the problems. Did we collect the right sample at the right time, stored it properly, did all the work properly? I think we can stop here and break and then. Yes.
Yeah. Only one question. So is that this high technology, using of high technology, somewhere which is misguiding the research to get a solution in an easiest way? So rather, we are really following the technology in a highest speed, rather understanding the consequences at the ground level. The best example is that in 1700, Edward Jenner, he discovered a vaccine, so which made to eliminate that pathogen, that is smallpox. So after 100 years, Louis Pasteur, he developed many vaccines. They work even today. They are working. But after that, so that kind of classical research is we don't find much. But now we have high technology. They didn't know anything about the cells. Even using the same microscope, they did all those things just by observation. Now we have excess of technology, but there is no that kind of breakthrough research. So somewhere we are really misguiding in the beginning of our research research journey. So now, if you ask, yesterday Ranjit Das said that now we need to have a genomic sequence data of a person to prescribe the medicine. Is that much is required to solve the problem? Say for example, best example, if you go to the two doctors, one doctor prescribe costliest medicine. If you go to the other doctor who is a service motto, he prescribed the medicine which is very cheap. The solution is same. So that's what I'm thinking. Is that much technology is really support us to find a good solution, which is affordable for everyone? This is very general question, just because the background is microbiology.
Right, so the question of how much does high tech help, especially if we cannot use it in everyone every time. So for example, for metagenomic sequencing, I think how it helps is that it tells us what else we can expect in a sample. Now you may not do metagenomic sequencing for everybody who comes. However, you now know these are the potential pathogens circulating in this region. Then you make the test for that. Those tests have to be cheaper, affordable, more specific, etc. And the more they can be point of care, the more they can be point of need, the better. However, in order to know that, I feel that technology can be a big player in broad scale understanding of things. Implementation of a high-tech technology everywhere is going to be a challenge. And in general, I don't think people try to do that also. Usually people do take the information and knowledge that you gain from something like this and find something that is more scalable. So it's a question of scale versus what you get out of it, right?
PCR, corona vaccine diagnosis. Now we stick on to the technology. Now it is very common, though it is expensive. People are trying to find out the alternative strategies just because it is available. Nobody is thinking which is expensive. It is better to find out the alternative which works in a cheaper way.
Well,
that's what I mean.
Sure. I mean, I don't have anything more to say.
Can I give example?
Sure.
Yeah. I will give example of polio vaccine. So we know that two different types of vaccines are there. One is oral and one is injectable. Injectable vaccine was developed using wild type virus. Dr. Salk has developed, and Dr. Sabin has developed attenuated vaccine. So to achieve attenuation, he has done many experiments. It was not easy for him to get that virus, 3 virus serotypes. Then later, we know that some issues are there with the oral polio vaccine, and now it is under eradication. So now with the help of technology, we can develop stable virus, which we can use as a vaccine. Like next generation vaccines are there, like S19, which we are new strain of polio viruses, 3 again. And we can use those viruses for developing inactivated vaccine and NOPV vaccines, novel oral polio vaccines. Again, example of technology which we are using to develop rapid vaccines and better vaccines, safer vaccines.